Welcome to our second session dealing with apoptosis and necrosis. As we already know, apoptosis can be introduced by binding of certain death signals to receptors outside of the cell or by cellular stress. So, what happens once a cell senses stress or a death signal is bound to a receptor? This leads to activation of certain downstream signaling cascades, and the most important players in apoptosis are so-called caspases. They are chief executioners of this very specific form of cell death. Caspases are cysteine aspartic proteases. So as a protease, they obviously cleave certain proteins. Caspases are a family of 12 proteases that exist as inactive proenzymes in the cell. And this is quite important concept in cell biology. Many proteases indeed are first secreted as proenzymes because cleavage of proteins needs to be tightly controlled. So in an inactive form, the protein is slightly larger, and caspases themselves need to be cleaved to be activated. So a portion of the protein which prevents the protein from being active gets removed. This leads to formation of an active caspase which is able now to cleave other proteins. So we are dealing with the so-called caspase cascade. There are two types of apoptotic caspase: the initial caspases and effector or executioner caspase, and this is something you need to keep in mind. However, both of those forms of caspases are present in the cell as proenzymes, so they themselves need to be cleaved to be active and to fulfill their role in the cell. As I mentioned before, caspases are arranged as a cascade. So once a molecule of caspase is activated by cleavage, this molecule can now cleave many molecules of the next caspase in the cascade. Once they are activated, they can cleave cytosolic proteins and possibly even more of molecules of the next caspase in the cascade. And finally, those caspases then can cleave proteins within the cell. Again, our initiator caspases activate other caspases. Then, effector caspases break down cellular components such as the cytoskeleton or the DNA. As you can see here, this whole process seems to be quite complex, but you don't need to keep in mind all of those caspases that I'm showing here on the slide, but you need to remember the concept. All caspases in our cells are initially inactive because they are present as proenzymes or procaspases. Once a molecule of caspase is cleaved, this results in an active caspase which can then activate subsequently other procaspases in the cell. However, in this process, there is one point of no return within apoptosis where apoptosis can no longer be stopped, and this is usually activation of caspase 3, which is the effector caspase. What you need to remember though is that in case of extrinsic pathway, the first caspase which is going to be activated is the receptor-bound procaspase 1. Once this procaspase is activated, this leads to formation of the active caspase 8, which in turn activates caspase 3, 6, and 7. Once those caspases are activated, this represents the point of no return in apoptosis, and cells die in a very controlled manner. In reality, this is slightly more complicated because there is a certain amount of crosstalk between those caspases in the cell. There are also molecules that are able to stop apoptotic cascades unless the point of no return is reached. But this is just to show you the big picture. Also, in terms of ligands that induce apoptosis, there are plenty of different molecules that can induce this process. For example, tumor necrosis factor alpha, we have heard about this protein already. This is actually a pro-inflammatory protein. However, if it binds to certain receptors, for example, tumor necrosis factor receptor 1, this leads to activation of the receptor-bound procaspase 8 and procaspase 10. Similarly, Fas ligands, which are quite classic activator of caspases, lead to cleavage of the receptor-bound procaspase 1, and this activates the subsequent caspase downstream. So, how does it work for the intrinsic or mitochondria-mediated cell death? This pathway can be activated by a large variety of cell stresses, such as free radicals present in the cell, DNA damage, viral infection, or absence of survival signals. Again, this is something we have learned in our cell fate lecture. If no survival signals are present, cells usually activate apoptosis unless we have to deal with cancer cells. The most important protein playing a role in the intrinsic activation of the cell death is cytochrome C. And Dr. Cottrell is going to be talking about this very molecule a lot in his biochemistry lecture dedicated to mitochondria and ATP synthesis. This protein is essential in the electron transport chain in mitochondria. And this is the process how ATP is generated in our cells. If the intrinsic apoptosis pathway is activated, pores form in the outer mitochondrial membrane, allowing release of this molecule into the cytosol. Once cytochrome C is released into the cytosol, it binds to other cytosolic proteins and forms a multi-protein complex referred to as apoptosome. And what you can see here is a crystal structure of an apoptosome. So as you can see quite easily, this is highly regular and really nice looking protein. The formation of the apoptosome requires cytochrome C, which is released by the mitochondria, but also a few other proteins that are present in the cytosol, namely a protein called APAF1, procaspase 9, and this also requires ATP. There are 7 molecules of each protein in the complete apoptosome, and this is quite large protein with a combined molecular weight of 700 kilodaltons. The end result of the formation of the apoptosome is the cleavage and therefore activation of the procaspase 9 into active caspase 9, which is an initiator caspase for the intrinsic apoptosis. So again, if the extrinsic or receptor-mediated apoptosis is activated, this leads to cleavage of procaspase 8. In contrast, in the intrinsic pathway, procaspase 9 is activated within the multi-protein complex called apoptosome. Apoptosis, especially the intrinsic apoptosis, is also tightly regulated by a family of proteins called BCL2. But again, this is additional information you don't need to remember for the exam. But please be aware that this process exists. The BCL2 family is responsible for the regulation of cytochrome C release from the mitochondria. There are around 20 members of this protein family, and they consist of both pro-apoptotic and anti-apoptotic proteins, allowing apoptosis or intrinsic apoptosis to be tightly regulated. Those proteins have the ability to induce apoptosis or prevent it, And this depends between the balance of expression of two types of BCL2s. As you can see here, there are plenty of pro-survival BCL2 protein family members, but there are even more pro-apoptotic proteins. Anti-apoptotic members of this family exist in the mitochondrial outer membrane and act to block the action of the pro-apoptotic members. The pro-apoptotic members of the BCL2 family work by inserting themselves into the mitochondrial surface and promoting the formation of large pores in the outer membrane. Once those pores are formed, this leads to the release of cytochrome C. Cytochrome C then can bind to cytosolic proteins, form the apoptosome, which leads to activation of the procaspase 9. So once apoptosis is triggered, this leads to activation of the caspase cascade. This in turn leads to K+ efflux into the cell, and this is something quite important because this does not happen in necrosis. Once caspase cascade is activated, this leads to surface blabbing and phosphatidylserine exposure. We have GSH extrusion, chromatin degradation, and cleavage of proteins called integrins. K+ efflux leads to cell shrinkage. Surface blabbing results in phagocytosis by macrophages. GSH excretion leads to oxidative stress. Chromatin degradation mediates nuclear fragmentation, and the cleavage of integrins leads to loss of cell-cell interaction. Those are the processes from biochemical point of view. At the cell level, even normal cell activates apoptosis and the caspase cascade is initiated. This leads to shrinkage of the cell and condensation of the chromatin. Then the membrane starts blabbing and organelles disintegrate. Nucleus and organelles collapse. The membrane continues to blab. Afterwards, apoptotic bodies form, and macrophages then can take up the apoptotic bodies. So, what you need to keep in mind here, and this is something very important because we could ask you this in the exam, what are the hallmarks of apoptosis? First of all, apoptosis is always characterized by cell shrinkage. Apoptosis maintains organellar integrity, and it also maintains ATP level. In apoptosis, ion homeostasis is kept. However, membrane surface starts to blab. Nuclear chromatin condenses and it's also fragmented. Apoptosis requires synthesis of death effector proteins and can be prevented by blocking steps in the death cascade. Apoptosis also does not adversely affect neighboring cells because the result are apoptotic bodies, which are vesicles that contain components of the cell. Importantly, the result of apoptosis is usually no inflammation. So with this, I'm closing the second session and I will see you in session number 3.